I'm located at the Research Institute Sintef in Norway, and I'm also past president of CRS. I would like to welcome you all on behalf of the board of the CRS focus group, Nanomedicine and Nanoscale Delivery, to our first webinar with the topic Nanomedicine Development Lessons Learned, Approval Earned. Before I introduce our speaker, I would like to give you some organizational information. We will have a lecture of approximately 40 minutes, followed by a Q&A session of approximately 20 minutes. You can send your questions during the whole presentation to me in the chat room, but we will not answer them before after the talk. Just a short question. I would ask everybody to turn off their microphone when they are not talking, because there is a lot of background noise at the moment. But now let me introduce our speaker of today, Dr. Jeffrey Clogston. Dr. Clogston is a principal scientist at the head of the Physicochemical Characterization section at the Nanotechnology Characterization Laboratory. In his position, he conducts physicochemical characterization and standardization of nanoparticles, develops new analytical methodology for critical quality attributes, and assesses current instrumentation for nanoparticle characterization. His areas of expertise include physicochemical characterization of and in vitro release from lipid-based drug delivery systems, analytical methodology, and protein and lipid biochemistry. And since the Nanocharacterization Laboratory has characterized more than 400 nanomedicines of all platform types, he has a lot of experience in this area. So, Jeff, the floor is yours.
Thank you very much, Ruth, for the kind introduction, and thank you again for allowing me the opportunity to speak with you today. So before I go into the bulk of my presentation, I just want to spend a couple of slides on the who and what of NCL, basically what we do. So the Nanotechnology Characterization Lab, or NCL for short, was established back in 2004 as a collaboration with NCI, NIST, and FDA, with 4 primary objectives. The first is obviously to characterize nanoparticles using standardized methods, to conduct structure-activity relationship studies, to help with the regulatory review of nanotech constructs, and finally to engage in educational and knowledge-sharing efforts like I'm doing today. So our clients involve government, pharma, academia, and biotech companies, and they submit our nanoparticles to us, in which then they are subjected to our assay cascade, which includes the characterization technique shown here that I'm using with my mouse. And I'll actually go into this in a little more detail in my next slide. We also do R&D for other nanomaterials. So these will include things like formulation, bioanalytical assay development, mechanistic studies. And the ideal really with any of these 2 types of characterizations or submission is to basically support our client in their development, clinical, regulatory, and commercialization needs. And if you take anything from this slide, there are 2 things. The first is our main mission really is to help accelerate the translation of promising nanotech and diagnostics, meaning get you from the bench to the bedside by providing preclinical characterization. The second point that you should remember is that our assay cascade is a free service. All one needs to do is apply to us through a white paper process where it will be reviewed internally, and if accepted, will then be subjected to this assay cascade for free. So if you want more details, please visit our site shown here. Now this assay cascade is a 3-tier assay cascade. The first tier is physicochemical characterization, which I head up. And in this section, we look at size, distribution, composition, purity, surface chemistry, stability, to name a few. And I've listed a couple of techniques associated with these parameters. We have an in vitro characterization group, in which case we can look at sterility, cell uptake, hematology, immune cell function, and toxicity. Finally, we have an in vivo characterization. So this will look at the the pharmacology, immunotox, toxicity, as well as efficacy. And the idea behind all of this really is to, okay, my screen is going, okay, there. My screen basically is to link all these physicochemical properties to the biological outcomes. Now, this will serve as my, okay, my presentation, okay, there we go. Sorry, having, let's see, there we go. So this will serve as my NCL summary slide. So we support preclinical characterization, we look at regulatory concerns, we do clinical characterization, we can explore alternate indications, as well as help characterize with next generation nanoparticles. We have 14 collaborators that are actually working the clinical trials with novel nanomedicine therapy. So I urge you guys to look at our website. So I want to begin now the bulk of my talk, and we'll begin this by defining what a critical quality attribute is. What I've done here is I've taken a definition as provided by FDA, and the reference is down here. I'm not going to read this to you, but in other words, basically, these must be clearly defined along with any of the potential risks due to changes in that attribute. They can include anything about the nanoparticle, the active drug, components, drug load, excipients, things like that. You want to, when you define these CQAs, or critical quality attributes, you want to consider the intended use and the route of administration. And finally, these are unique to each formulation. That is, you have to specify the key CQAs for your given formulation. So how do we do this? So typically, CQAs will be physicochemical properties, such as the ones I've listed here. And what you want to do basically is try to determine which of these, if any, will affect a biological outcome. For example, the PK or biodistribution. Now this seems very daunting and very challenging, and it is actually. Try to identify which is the CQA as well as how to measure that is very challenging. So let me just give you an example here. Our client submitted to us, these are pegylated colloidal metallic nanoparticles. 2 batches were submitted. In the first batch, when we did tox studies, we saw extensive lung lesions, whereas the second batch, we saw nothing, basically. It was largely benign. And we measured the size, charge, and polydispersity of these 2 batches, and they were basically the same. So the question is, well, obviously, the CQA are not size, charge, and polydispersity, even though these are probably the most basic and fundamental parameters one would measure. So the question then is, how do we define or figure out what these CQAs are? And I'll come back to this example in a couple of slides. So how do we identify CQAs? Well, the easiest way to do this is to use knowledge of prior drug products. So if you worked with similar products, see what they use as their CQAs and check those. Do lots of testing on multiple batches. You want to assess limitation with respect to polydispersity, concentration, etc. So you want to look at the high and low of each of the parameters. For example, size. You want to look at the low end as well as the high end and see how that affects your biological outcome. You want to verify relevant in vivo biological testing, and obviously you want to monitor these as you do them in process. And as I mentioned before, these are very hard to do and difficult to define because of the complexity of nanoparticles. So what I'll do now is I want to give some examples of how we can define critical attributes. So here again, I go back to the sample example where we had the pegylated colloidal metal nanoparticle. In this case, what we did was simply centrifuge a sample to isolate the pellet, which contains the nanoparticle. The supername contains the PEG, and then we assay both batches for presence of PEG, both in the supername, which would represent the free PEG unassociated with the nanoparticle, and the pellet, which would contain the PEG associated with the nanoparticle. And what we saw that there was actually differences between the 2 batches, and this is what actually caused those lung lesions that we saw in the tox study. So for in this case, the surface ligand density is the CQA, and that is specific to this formulation. One thing we should always look at is multiple batch testing. So again, what I'll show you here is now kind of give you guidelines and tips on how to determine CQAs. So in this example, we have pegylated metal oxide nanoparticles, and our client gave to us 3 different lots. Now, usually for metallic nanoparticles, TEM is the way to go to looking at size. And that's what's shown here. You can see the TEM micrographs for all three lots. Now, I don't know about you, but if I look at all three of these, they look the same. And if you were to do size analysis on these three lots, they're pretty much the same. So obviously doing TEM is not the way to go to look at sizing for this sample. What we did instead was use AF4, which is asymmetric flow field flow fractionation, in conjunction with the multi-angle laser light scattering detector, and we ran that through, and that's what's shown here. These traces are the light scattering signal for each of the different lots, and across each of these peaks is the radius of gyration distribution or size distribution. And it's obviously clear from here that lot 1 was different from lots 2 and 3. So again, trying to figure out what technique is very challenging in this case, where the usual traditional TEM didn't work. We had to resort to a different type of measurement using AF4 mols. And it's usually a good idea to test multiple batches when trying to determine what your CQAs are, because if you just use one, obviously you can have false claims and irreproducible results. Another example of multiple batch testing is when our clients submitted to us 13 lots of starting material, in this case a G5 PAMAM dendrimer, and they were seeing different results with their final lead product. So we asked them for their starting material to characterize that. So they gave us 13 lots, ironically, or unfortunately on our end, to characterize. And with these 13 lots, we did multiple assays to look at differences between these 13 lots. This involved DLS measurements. We did HPLC to look at purity. We did molecular weight measurements using MALDI-TOF. We measured the zeta potential. And we did titration experiments to determine the number of amines on the surface. So based on this set of data, it was actually the purity by reverse phase HPLC and the number of amines by titration, which will give the differences between these 13 lots. So in this case, you want to tailor the characterization techniques to the nanomaterial. Now, some more guidance in regards to picking CQAs and in general is you want to make sure you include the proper controls when doing your analysis. So for example, if you're doing an efficacy study, you always want to include the standard of care of treatment with your sample, your nanoformulation. Really, this is a way to test if your formulation has any chance against the standard of care treatment. And really, you want to run this. You want to run the empty platform as well as a free API. These are more for looking at background interferences and things like that, or unrelated tox. If you have a targeting formulation, you want to make sure you run the non-targeted. Therefore, you can prove that the targeting actually works. And this is really important. You want to compare at equitoxic and equal doses. This refers back to the standard care treatment. In the example here, they're using Taxol as their standard of care. You want to run your formulation, in this case the micelles, at the equivalent equitoxic dose to make a fair comparison. Continuing with the control topic, you want to make sure the proper controls are run. So again, this could be the blank or empty formulation. These would be the precursors in your synthesis steps, things like that. Because a lot of times, nanoparticles may interfere, inhibit, or enhance some in vitro assays. So for example, here we have basically endotoxin testing. If we spike endotoxin in water, we can measure the amount, and it's 0.4, which is the same as how we spiked in. If we have that spiked into gold nanoparticles, we can see that the signal is actually suppressed or inhibited. If we use polymer nanoparticles, we can see that this is actually enhanced. So you want to run the proper controls when looking at certain assays. The next topic has to deal with testing in biological matrix. So most people will test in buffer or what have you, and there's nothing wrong with that from an R&D point of view. But if you want to look at translation of your nanoparticle, you want to test it in biological matrix. So this would include, blood, whole blood, plasma, serum, whichever you can get a hold of and actually use in your equipment. And the reason is because they may become unstable in biological matrices, they may precipitate, and that's what's shown here. A lot of times nanomedicines, if you're using polyplexes or prodrugs or micelles or emulsions, things like that, which are held together by non-covalent interactions, once you put this in a high ionic strength, it may fall apart. Finally, you want to look at the stability of your nanoplatform when you put this in plasma. Sometimes the drug will release too fast or too slow. in which case then toxicities are similar to that of the free drug. I kind of like this, my colleague Steve Stern, our toxicologist, came up with this little diagram here. I like this a lot because really it highlights or summarizes what you should look for in terms of stability of your formulation as it pertains to drug release. So if it's too stable, the release will be too slow, you have bioaccumulation, it doesn't work, it's non-efficacious. If it's unstable, it will release the drug prematurely, you have off-target toxicity because of this, and it's not optimized. So there's this fine balance between being too stable and unstable, and it's called ideal stability, which you want to try to hone in on. So how do we do this? I mean, it seems like a lot of different things to look at. So as a starting point, the historical PC parameters to monitor include polydispersity, so looking at sizes. So you want to define upper and lower limitations on the size and shape populations. You want to look at drug loading. If your particle contains multiple peaks or multiple size fractions, you want to look at the drug distribution across these size fractions. You want to look at surface coating. Like in my example before, how much of the surface coating or ligands are actually bound to the surface of the nanoparticle, how much is free. You want to look at density and orientation. Drug release, and I'll get into this at the very end of my talk. You want to measure not only drug release, but you want to know how much of it is actually free, how much is nanoparticle bound versus protein bound. And like I mentioned on the previous slide, you want to do this in a biologically relevant matrix. You want to look at impurities, so surfactants, things that you use in your processing. You want to make sure they're not present in the final product if they're toxic. You want to look at acceptable levels of excipients as well. Stability, like I mentioned before, you want to look at that ideal stability, that nice balance between being too stable and unstable. And for this, you want to basically use multiple techniques for each of these parameters, repeat on multiple batches, and then this will basically give you your release lot specifications for your product. So we looked at PCC. I also want to look at just biological properties as well. I'll plead the fifth here because I'm not an immunologist or a biologist. I'm a chemist by training, so I won't know the details of this, but my co-worker, Marina, has published 2 papers here which details all of this information, as well as this nice little tree that she came up with. It's a lessons learned structure-activity relationship, and it's concerned with endotoxin testing, cytokine storm, hypersensitivity, complement, thromboticity, etc. So I urge you to look at this if you're looking at biological properties to monitor. So now I'm going to kind of switch over and focus on the physicochemical characterization, as this is my area. So if we think about physicochemical characterization, it really boils down to the analytical instrumentation and development of new methods. So in-house, we have to our disposal all these different techniques. Now, I've been here for a little over 13 years now at NCO, and within those 13 years, we have been able to identify the critical quality attributes and methodology needed to characterize many different platforms. I listed 3 major types of platforms here: liposomal products, polymeric nanoparticles, and colloidal metal nanoparticles. So what I wanted to do was to take all that experience and try to condense that into one slide, which is very challenging in of itself. So what I did was I used the FDA liposome drug products guidance for industry basically to guide me. And within this document, there is a section called the CMC, the Chemistry, Manufacturing, and Controls. And within this, there's a critical quality attributes. So what I did was go through this section and basically, with my experience and NCO general knowledge, came up with what I call a cheat sheet, and that's given here for liposomal products. I call it parameters, methods, and considerations. And what I've done here was basically list the key parameters, so size distribution, morphology, surface characteristics, composition, purity, stability, batch to batch, as well as starting material characterization. Now these can be CQAs depending on your formulation. This is a good starting point when you're trying to define what a CQA is for a given product, if it's liposomal in nature. For each of these parameters, in the case of size distribution, I give the more common techniques along with examples on how to measure it. For the other ones, shown here and here, what I've done is I didn't give you techniques because there are many ways to look at this, but instead I've given you things to question or things to think about. For example, if you look at composition, you have drug concentration, total, free, encapsulated, drug distribution, etc., and I give you examples of some of these as well for each of these different parameters. So this is actually a nice cheat sheet for you to look at if you're working with liposomal products. Now, what I did then was I took this rationale and this line of reasoning to look at polymeric nanoparticles. So you'll see the same parameters here, but now the methods and the questions will be slightly different as it pertains to a polymeric nanoparticle. And again, I give you examples. And I took this one step further, and I did it for colloidal metal nanoparticles as well. And you can see there's a lot of examples here. So this is actually available to you guys. If you're interested, just shoot me an email. If you're interested in this, my email is on the title slide. It's also on the final slide, and I'm sure you can get my contact information after the talk if you're interested in these three quote-unquote cheat sheets. So what I want to do now is kind of give you some examples or some really cool techniques that we have devised here at NCL regarding these three cheat sheets. So the first example that I have, and this is really not an example, but more of a lesson here, is sizing by different techniques. So our clients supply to us a lipid protein nanovesicle, and we did DLS on this. As you can see, it has a size population around 100 nanometers or so, and there's some stuff out here, but as you may know, DLS is not suitable for particles greater than 1 micron in size. For that, we looked at laser diffraction. So laser diffraction can pick out this larger size population as well as a smaller size population. And then we looked at a third technique, and I mentioned this earlier, AF4, asymmetric flow field flow fractionation, which actually separates the nanoparticle into different components based on size. And using MALS, multi-angle laser light scattering, UV, as well as dynamic light scattering detectors in line, we can actually identify and size each of the different components in the sample. So the take-home message here really is that orthogonal methods are highly recommended. So if you can, it's always good practice to measure, for example, taking size as the simplest parameter, size in many different ways under different conditions, and it's also important to know what you're measuring for that given technique, right? So for DLS, it's a hydrated size, for example. The next thing to look at is particle size and concentration. So here we're using resistive pulse sensing. I'm not sure if you guys are familiar with this, but what this really does is you're now looking at sizing on a per particle basis, and I think the field itself is moving more towards that as opposed to batch mode measurements or ensemble methods such as DLS, which are averaged across all the size populations. So here's just an example where we're taking 3 commercially available nanoformulations, liposomes in this case, Doxil, Sunpharma, and Onivide, and we're just simply measuring the size distribution as well as the concentration, that is particles per mil, for each of these 3. Now this can be used again as a CQA if this is relevant. I like to show the zeta potential slide here because I think everyone has done zeta potential. Usually when I give this talk in person, I will usually ask the question to the crowd, with a show of hands, how many people have done DLS? And, more than half the crowd usually raises their hands. And then I'll say, with those raising your hand, how many have done zeta potential? And usually the same people will keep their hands up in the air because it's the same instrument, right? So, but I do want to point out that zeta potential, if you're going to use this as a CQA, zeta potential is really dependent upon the measurement condition. So depending on the pH of your sample, you can change the zeta potential of your sample. The ionic strength of dispersing media, if I change the ionic strength here, I can change the zeta potential of your sample. So the take-home message here really is you want to report the conditions that you make your zeta potential measurements at. And if you're using this as the comparative method, you want to make sure they are consistent across the board. And the last thing to keep in mind is that, zeta potential, because it's also based on light scattering, there is limitations in resolving power, so just be mindful of that. I always think of zeta potential, if you're familiar with Z average in DLS, it's very similar to that, where you're getting average zeta potential across all the species in your sample. And I have a paper listed here that we published about 2 years ago which goes into more detail, the zeta potential, as well as with liposome characterization. Another CQA can be looking at lipid composition for nanoparticles containing lipids. In this case, our client submitted to us 3 batches of lipid nanoparticles with siRNA encapsulated within the interior, and they wanted to know basically batch-to-batch consistency, so we came up with a method to look at the lipid composition, that is individual lipid composition, and we came up with a reverse phase HPLC with a charge aerosol detector to look at this, and we can actually see all 6 lipids and quantitate the amount, and in this case all 3 were the same. So looking at from that point of view, we can use this to assess batch-to-batch consistency. I do want to point out, for those of you who work with liposomes and are into, looking into individual lipid composition, the HPLC method that we have here, or that we use for this, will probably work for about 95% of all liposomes you work with. If you're in the field of liposomes, typically liposomes are made of 3 or 4 lipids. You may run into some proprietary lipids, in which case you have more, but the solvent system and gradient dilution and whatnot, column conditions, works great for most of the lipids that you would ever work with, and that method is actually given on the last slide reference there with the zeta potential study, so please look into that if you're working in this field. Another interesting is to look at protein binding. So this is pretty popular now to look at the protein corona, how much protein binds to the surface of your nanoparticle, because this will affect biodistribution. And what we've done here is to qualitatively assess protein binding by using AF4 in line with mols and DLS. So the AF4, like I mentioned earlier, will separate the nanoparticle from the protein. The mols will actually measure the radius of gyration. It's a sizing measurement. And the dynamic light scattering measures the hydrodynamic size. And to demonstrate the utility of this method, we will look at 3 commercially available liposomes. The first one is doxyl. So doxyl has about 5 mole percent lipid PEG. If we look at onivide, it has some lipid PEG, but not a lot. And finally, if we look at ambisome, this has no PEG at all. So simply what we do is we take each of these formulations and we run the sample with and without the presence of human plasma, and we run it through this setup. And let me walk you through here. So shown here for doxyl, for the first case, we have the AF4 mol. So this is the light scattering based off of mols. And in blue is basically the before, and then the red is after incubation with plasma. You can see that the AF4 separation can actually separate the proteins from the nanoparticle, and then this is the DLS. What we're looking for here is the ratio of Rg over Rh before and after. As you can see, they're the same, so this implies that there's no shift in the mass distribution, indicating minimal protein binding. If we look at onivide, onivide, you can see now that ratio has increased, implying that there is some protein binding to the surface, and this is most likely due to the lower amount of PEG in the formulation. If we look at ambisome, which has no PEG, this ratio actually almost increases by 50%, signifying significant protein binding to the surface. Now, you don't even have to measure this. You can just look at the fractogram to show that something is obviously binding to the surface of the nanoparticle. So this is actually a very quick way to assess protein binding, as well as size distribution in the presence of plasma proteins. We are currently writing this methodology up for publication. Further with the whole idea of looking at doxyl, because a lot of people would work with doxyl and doxyl analogs in the market these days, going beyond size, charge, drug loading, things like that, one thing we can look at is ion quantitation in them. So if you're familiar with doxyl, doxyl is actually a precipitate, a crystal within the interior of ammonia and sulfate. So we came up with a method where we can actually quantitate the amount of ammonia and sulfate in the formulation. Not only can we quantitate the amount of it total, using microcons we can measure the external, and then through mass balances calculate the internal, and then you can use that as a check to make sure that this precipitate, the stoichiometry, is correct in your formulation. This actually supports FDA regulatory guidance for such injection products. You can use this for batch-to-batch consistency as well as doxyl analogs. You can use this for other counter ions in the formulation as well as buffer ingredients. And we recently published this methodology last year, or actually this year. My final example with liposomes deals with impurities and stability assessment. So again, we'll use everyone's favorite liposome, doxyl. So if we look at doxyl, it has the following components: HSPC, DSPC, MPEG, and cholesterol. If you look at HSPC, hydrogenated soy phosphatidylcholine, it's actually a mixture of DSPC and PSPC or SPPC. All these acronyms. What you need to know basically is that with time, these will hydrolyze and give you free fatty acids, namely stearic acid and palmitic acid, so your C18 and your C16. So we came up with the reverse phase HPLC method using CAD detection to quantitate the amount of free fatty acid as a function of time for several lots of doxyls. So we just happen to have 4 lots of doxyls with different expiration dates dating back to 2011 and as most recent as 2018. And as you can see, with time, more free fatty acid evolves. And you can see that here clearly as this peak increases. We can also look at the increase of the lyso product here. So this is one way to assess degradation products in liposomes, and this can be used to assess stability and shelf life of your formulation. Now I'm going to switch over to polymeric micelles quickly here. In this case, a lot of people use prodrugs, and you want to look at the drug release in prodrugs in a biological matrix, right? In this case, human plasma. So we had submitted to us a prodrug loaded micelle. So there's a prodrug in which they took a drug, they used a linker to conjugate it to a lipid anchor, which then, with other components, formed your micelle. So the question was, well, does it cleave, first of all? How fast does it cleave? Where does it cleave? And is it still active? So in this case, we came up with an HPLC method where we took the prodrug intact and monitored basically it as a function of time after incubation with plasma. So at time naught, you have, basically your prodrug. With time, as this hydrolyzes, this peak gets smaller as it basically gets hydrolyzed. And then you can use this data to come up with your release kinetics or hydrolysis rates of your prodrug. This case was interesting, and I kind of, most people look at the drug as it's cleaved off. The problem with that is if you're working with plasma, there's, esterases, enzymes in there which will start breaking down those links. Depending on your drug, and I think most drugs have ester linkages within them, if you carry this out for more than 2 days, your drug itself will start basically falling apart. And then trying to do a mass balance on all those components is a real pain. So sometimes if you're working with a prodrug, a better approach, or an easier approach actually, would be to look at the intact prodrug as a function of time. We then did LCMS to look at basically where this cleaved and if the drug was still active. Another example is to look at drug stability of polymeric nanoparticles. And again, you'll notice I use a lot of these AF4 techniques. In this case, we're using AF4 with DLS in conjunction with HPLC. So our client submitted to us a polymeric micelle, which had drug encapsulated. We did a batch mode measurement of DLS, and you can see it's polydispersed. The question then is, well, where is the drug located? Because this sample is really polydispersed. So using AF4, again, we can separate the particles based on size. We can measure the DLS of the particle. So you're getting size distribution on a fractionated sample. Okay, I lost my... okay, sorry about that. Having some technical difficulties. Where did my presentation go?
I can't see it either.
Where'd it go?
And
yeah, it kind of just disappeared on me. Let's see.
I wonder whether Bruno did something, because I didn't do anything, I think. Oh, here it is. Oh, here it is.
Yeah, it keeps... There it is. Yeah, I found it. Yeah, it keeps doing this on me every now. Sorry about that, guys. Every now and then it minimizes on me, and you probably see that. But anyway, let's go back to this. So using AF4 DLS in reverse phase HPLC, what we do here is as it comes off the AF4, we collect fractions and then assay that for drug content using reverse phase HPLC. So these are the results for this formulation. As you can see, here is the size distribution or the flow mode DLS results. So you can see there's two populations here. one a constant population of 55 nanometers and an increasing size population. What's interesting about this is when you collect the fractions and assay for drug, only 14% of the total drug was recovered. And the reasoning behind this, and if you're familiar with AF4 as a technique, one of the first steps you do in that method is to passivate the membrane with BSA, bovine serum albumin, a protein. In this case, the drug partitions to the BSA on the membrane. It has more affinity for the protein than a nanoparticle. Now, if you put that in a biological context, that's not a good thing, because that implies that your drug will be released right away when injected. And we followed up this result with the biological result. So my colleague, Steve Stern, has developed a method where you can actually look at the drug fractions, the different populations, bound, unbound, free, etc. And in doing that type of analysis with this formulation, about 85 to 100% of the drug was released within 10 minutes. So this coincides with our analytical method. Actually, I will talk about this method at the end of my presentation, as it's a very nice method to use, especially when you're looking at PK as it relates to generics and follow-on products. Moving on to, I have two examples for metallic nanoparticles. So this one, our client provided to us a gold nanoparticle coated with lipids and proteins. Since this is gold, you can actually dissolve the gold interior using KCN, and this essentially liberates the coating. You can look at the protein by fluorescence and the two lipids using reverse phase HPLC. So this is actually a nice little method if you're working with colloidal gold to look at surface quantitation. My last example is again with metallic nanoparticles. In this case, we're looking at a core, gold shell silica core pegylated nanoparticle. Our client gave us 3 batches to look at. In this case, we're using tunable resistive pulse sensing. This allows us to do size concentration, as well as size measurements and zeta potential on a per particle basis, and that's what's shown here. You can plot the zeta potential as a function of particle diameter and then use this to look at differences between the 3 lots. So with that, I want to move on to my last topic, and this is really how, measuring drug release. So a lot of people struggle with this and they don't know what to look at and how to do this. So we have developed several methods in-house. The first is a blood partitioning assay. I'm not going to go into details of the first 4 in the interest of time, but I do have references listed here for you guys to look into if you're interested in that particular assay. So we have blood partitioning, we have dual labeling. By this, you're basically labeling the drug and then some component of the nanoformulation to track them. There's extraction methods to separate free and encapsulated. These will be your SPE cartridges. You can do modeling, and there's a paper here by Steve Stern in our group that addresses this. And finally, there's a measurement of fraction unbound to quantify the amount of drug encapsulated versus released. And I actually want to focus on this, and this has been published as well. The reason I want to focus on that is as it pertains to bioequivalence or follow-on NBCDs or non-biological complex drugs. So therapeutic equivalence is defined as a pharmaceutical and a biological equivalence. Well, bioequivalence can be used in place of biological equivalence to study the generics, and then you assume that the PK represents equivalent biological effects. So if you can measure PK for your formulation, you're good to go. Now, obviously, that's pretty hard to do. It's not an easy task. And just some quick nanomedicine PK. It's much more complex than small molecules. So you have your nanomaterial first, which has drug encapsulated, and that's shown here. You then have this unencapsulated fraction, which consists of both your free unbound drug as well as your unbound, or your drug bound to protein. So the idea is to quantitate all 3 distributions for your drug. And Steve Stern in our group has come up with a nice method for doing this. It's actually, he calls it Situa. It's a stable isotope tracer ultrafiltration assay. And I'm not going to go into detail, but quickly what you're doing is you take a stable isotope of your drug and you spike this into your formulation along with the protein. You simply spin, centrifuge, and then you measure both of the stable isotope of the drug as well as a normal drug, and then using mass balances, you can come up with each of these 3 distributions. And these are summarized in the 2 methodologies and papers listed here. I highly encourage you guys to look into this. It's something that we work with a lot in our group, and it's a great way to really test your formulation against, let's say, the standard of care formulation. So this will be my last slide, and this will serve as a summary slide. And really what this is, it gives kind of like the rationale or the thought process of academia versus preclinical development. And it's basically like a guidance too. So like I mentioned earlier, you want to do multiple batch testing. You know, reproducibility can be a major issue. It's a good thing to test multiple batches and define your CQAs earlier on in the process. Scale up obviously is a huge issue. There's a whole field of scale up, right? You want to address this earlier on. So for example, if you're making liposomes with a 1 mil lipid extruder, right, syringe extruder, how are you going to translate that? You're not going to do that obviously by making like a liter of sample. It'll take forever. You want to look into more high throughput methodology. And you want to make sure that when you make a small versus a large batch, that it's reproducible, not only in PCC, but also in a biological setting. So you should probably do bridging studies to prove that the efficacy is still there. As I mentioned earlier, and we talked about this, you want to have informative control arms, you know, to make sure there's no false positive, negative results. The best way to do this is to have, you know, all the precursors, the empty, have your free drug, your non-targeted samples as well, and run that in parallel with your lead formulation. You want to do measurements in a biological matrix. Finally, there's predictive in vitro and vivo models out there that you can use. And the end point for all this really is you want to translate and apply to a regulatory institution. And because of this, the threshold is much higher. So it's important to address these issues early on. With that, I will leave it here, and I want to thank my group. And I want to thank you guys for listening, and I can entertain any questions now. Thank you.
Thank you, Jeff, for this really highly informative and interesting lecture. I have started to get questions, and two questions are very similar. They are about the best methods to quantify drug loading efficiency or to measure encapsulation efficiency in various types of nanoparticles.
Yeah, so that's a good question, and it's funny because I can always answer a lot of these questions well with it depends on your formulation, of course. And so I'll tackle both of these as one question. So polymeric and liposomes. So those cheat sheets that I showed earlier, actually you can use that. There's methodology there to look at drug loading and release kinetics and things like that. That method for Situa that I mentioned on the last 2 slides, I guess, that's a good way to look at release kinetics in plasma or serum. The best way I would do, you know, to measure encapsulation efficiency, well, normally if you're working with polymeric or liposomes, you're going to measure total loading first, usually through HPLC, right? With either UV or fluorescence, depending on the drug that you're using. That will give you an idea of total loading. The easiest way to look at free unencapsulated, you can use microcons. These are those centrifugal filters. Pick the right cutoff, molecular weight cutoff. You can run that through. And then the filtrate will contain any free drug. So that will give you an idea of the free drug in your formulation. Make sure when you're doing that, you run all the controls with that because you can have sticking of drug to the membrane, to the filter. It depends on the drug, obviously. And then because of that, you can actually isolate what's left as the retentate to look at drug loading there. It should mass balance out. There's just some ideals. It's a case-by-case study. If I didn't answer your question, whoever answered that question, please contact me and I can help brainstorm some other ways to look at it.
Another question that is quite related to that is what do you think is the best release medium, plasma or serum, when you are looking at the release of, for example, therapeutic protein as a prodrug?
Okay, so for that, I mean, I always, you want to go to the most relevant matrix. You know, if you can do it in blood, that would be great. But most people don't work with blood for many reasons. There's safety reasons associated with that. A lot of your instruments can't handle blood. You have to worry about things like that. So I would recommend plasma next because plasma will have all the esterases and things like that. It's a little more messier than serum in that case because you still have the clotting factors in there. Either one works. I tend to go more towards plasma, but definitely pick the one that you have access to, obviously, and that your instrument can handle. You're not going to, for example, AF4, you're not going to inject blood into it, right? So I would urge to use plasma at a minimum. Buffer is okay when you're doing R&D. If you're just looking initially, what kind of linker works at this pH, sure, use buffer, But then ultimately, once you decide on a formulation or a prodrug with a certain linker and a certain release kinetics associated with that linker, I would do that in plasma.
There are another question is you were talking about cytopotential measurements. What is the maximum mean particle size recommended for accurate cytopotential measurements?
Sorry, I'm saying.
Yes.
For zeta potential? Sorry. What's the size for zeta potential?
What is the size limit? The maximum. The size limit. The maximum mean particle size. So you get reasonable cytopotential measurements. I think is the question.
Yeah. Okay. Yeah. For zeta potential, it's based off of light scattering, right? Depending on what instrument you use. You can measure into the microns with zeta potential. I've done that with our instrument. I've actually looked at polystyrene beads, up to 10 micron, and measured the zeta potential of those. They're negative, obviously, because they're carboxylated. But yeah, you can get into the micron. Now, hopefully, usually I don't get that question because usually if you're working in a nano field, you're more on the other end of the spectrum, more on the 100 nanometers and that thereabouts. So, but yeah, zeta potential can work for larger size, micron size particles for measuring zeta potential.
Another method-related question is whether AF4 is suitable for assessing particle size distribution of micelle-based systems or systems that are sensitive to dilution.
Yeah, that's a great question. So in the past, people say, everything you can work with AF4 because AF4 is a gentle separation technique compared to, let's say, using size exclusion, for example, and separating things. So AF4, we can use it for micelles. We've done that, and actually that one example I showed was a micellar product. But that's a good question because we have run into instances where if you take a formulation and you dilute it too much, it falls apart. So for example, a Braxane, you can't run AF4 on a Braxane. So if you're familiar with a Braxane, it's basically human serum albumin with paclitaxel. And the idea is that when that gets diluted, it falls apart in the body. Well, in the AF4, if you're familiar with it, there's a focusing step. And during that step, you can actually, your sample is subjected to high amounts of diluent or solvent. And essentially, the Braxane falls apart. Genexol falls apart under those conditions, which is actually a micelle. So yeah, there are concerns to analyzing some of those samples by F4. I mean, for the most part, I would say 90% of samples work AF4, and there's a handful that we've come across where it doesn't work, but that's a good thing to look at. The easiest thing to test is to do DLS initially, batch mode, as a function of dilution and see if it falls apart. If it falls apart at 1,000-fold, for example, dilution, then it probably is not going to work on AF4. Again, these are just guidelines. It really depends on your particle, and I urge you to contact me and we can discuss more, and I can give better insight or guidance once I know what you're working with.
Another question that came in now is whether plasma protein, like albumin, will mask the released prodrugs lower in molecular weight than the plasma proteins.
So plasma proteins mask the released prodrug. It could, but so what you normally do is, in our case, it's incubated with plasma. You're going to use some kind of workup on your sample. So in that case, you're going to precipitate out the proteins initially by using cold acetonitrile. And then that will precipitate out the protein, and your organic phase will contain your drug. And then you assay that by reverse phase HPLC, in which case there's no more proteins there.
Another method-related question is whether it's okay to measure the cytopotential in deionized water, or what is the minimum amount of salt needed for zeta potential measurements?
So that's a good question. So ideally, when you do zeta potential, you want to have some ionic strength because you are looking at the movement of ions. So I would urge not to use water in that case. You want to have some ionic strength. Now, ideally, if you can measure this in PBS or saline, that would be great. But from an instrument point of view, that's very hard to do because if you imagine you're applying a high voltage to a high ionic strength media, you could essentially burn your sample in doing so. So what we do in practice is actually measure at 10 millimolar sodium chloride. So it's high enough where you have some ionic strength, but it's not too high where you risk the sample degrading with high voltages applied. Now you can take any measurement you want. I know NIST, for example, uses 2 millimolar sodium chloride. It really doesn't matter, but just pick one and be consistent in all your measurements for comparative reasons. If you do measure in water, keep in mind water will give you the highest value. As you add or increase ionic strength, your zeta potential will decrease. And that really goes back to how zeta potential is defined. Read that paper that I cited on one of my slides. It goes into all that, actually.
Another question is, how do you select the positive and negative controls for nanomedicines, for liposomes or polymeric particles, especially maybe in vitro assays?
Yeah, so the controls, it depends, right? So, again, it depends on the nanoparticle, what controls you want to run against your formulation. Like I mentioned on the earlier slides, if at all possible, always run your precursors if you can. Run the empty, run the non-targeted version of it, things like that. Run the free drug. With the free drug, though, a lot of times if the drug is not water soluble, you have to bring this up in DMSO. So you'll have to run a control of just, that percentage of DMSO in buffer to make sure DMSO isn't causing the effect. You want to run that. You want to run the appropriate standard of care. That is the one that you're trying to compete against. It really depends on your formulation, so it's kind of hard to, I'm just giving general guidance. If you want more specific answers, just contact me and I can discuss that with you.
Another question that came in is whether enzyme activity can be different in plasma from different species, like esterase activity in human plasma compared to mouse plasma.
Definitely, definitely. We see that in a lot of our met situa met that I mentioned. Steve and them actually sees differences because, the enzyme profile and activity for them are different between species. I tend to always use human plasma because ultimately that's what you're going to and you want your formulation to go into. So I would use human plasma first, but I understand sometimes it's not easy to get human plasma and you have to use either rat, mouse, whatever. I would test multiple, to be honest, just to make sure that it's consistent if you can't get a hold of human plasma.
One of my own questions is when should a nanomedicine developer contact the regulatory authorities at the first time? What is your experience? Should we do that during the discovery phase already, or when the CQAs have been determined?
So there are pre-IND meetings that you can have with them, where you can sit down and basically say, we have this, and they can give you some guidance early on. Some of our clients actually do that ahead of time, right? It's still a meeting, but it's not a formal meeting. So once you submit an IND, that goes into their records and archive, and they have that on your formulation. But with a pre-IND, you can go in there and kind of pitch your ideal to FDA, and they can give guidance. I think that's a good thing to do early on, obviously, because they can help guide you toward the right direction. So I would encourage that. Now, obviously, you can't go too early. You have to have some data, right? You should have some efficacy data, some idea of what your drug is, how you're going to encapsulate this or deliver it using nanotechnology in this case. But minimum have efficacy data for a formulation that you think is the lead, and then go to FDA as a pre-IND meeting to get information from them.
Thank you. I got another question, and that's what's the best method to measure the size stability in plasma?
That's a good question. So, there's a lot of publications on this, how to make measurements, and so most people will use DLS, but I should caveat that because depending on your formulation and its size, if it's the same size as protein, then you're not going to see it. If it's too small, you might be swamped out by the protein signal. If you're larger, then you might be okay. Usually what I do, to be honest, I use a lot of AF4 methodology. So you can incubate your sample in plasma and then run AF4 with DLS and mols in line to look at size stability as well as drug stability in the presence of plasma proteins. So these days within my group, we don't do batch mode. We still do batch mode DLS as a quick way to check the size. But really, we want to look at a flow mode where you have some kind of separation upstream, and then you look at the DLS sizing of a fractionated sample. So I recommend that in the presence of plasma because it works for that. There are other methods out there, obviously, but that's the one that we use in-house the most with the best results.
Another question that came in is, what is the ideal volume as a release medium in terms of plasma? When you do a release in plasma?
Yes, that's it. Yeah, so typically, again, if I go back to looking at, let's say, AF4 as a method to look at plasma, whatever. So normally what I'll do is I will incubate the particle with plasma. The plasma will be in excess, way excess. It's usually like in excess of 10-fold relative to the particle, right? And then once you have that, then you have to dilute the sample accordingly for your instrument. So for example, if I'm doing AF4, I can take a sample, incubate with plasma, let's say 1-to-1 in volume, right? Whatever. And then that would give me a 50% plasma. I have to, because of the way AF4 is set up and the plumbing and the diameters and things like that, I have to dilute down to 10% plasma before I can run that into the system. And that's how I have to run those samples, just because physically it will clog the plumbing as well as foul the membrane. But if you're using, for example, Steve's method, the Situa method, I know those guys, when they do their incubations with plasma, I mean, it's in excess of 10-fold of your nanoparticle. Because in their case, it doesn't matter. They're going to ultimately just centrifuge this out anyway. So in that case, it doesn't matter. But like I always say, it depends on the formulation and things like that. If contact me and I can give you more details and better guidance.
One last question. What are the most common pitfalls that you have met in your 13 years?
Pitfalls? In terms of characterization or just PCC in general or things like that?
I think both in characterization but also in samples that you have got. When are the what are the biggest hurdles where people fail?
So yes, I think in terms of, like in general, let's say, formulations that come in that fail, I think what you need to do earlier on, let's take an academic setting, for example. Usually in academia, we get submissions from a group that's chemistry heavy or biological heavy. And that's great because they know their stuff in their field, but they don't know the other field, right? So you can have a chemistry group with this awesome linker that does this and that, but they have no way to apply that in a biological setting, and vice versa, right? I think early on it's good to have a group or work with a group of people who have background across the board so you can see different point of views. I think that's important when you're formulating or coming up with a new formulation, and I think that helps a lot with the characterization as well. And I should make mention, because I have the slide up here right now, you'll notice our group, that's what we have here at NCL. We have PCC people, chemistry people, we have immunologists, we have pharmacologists, toxicologists. So we have the whole board, so to speak, to look and characterize a nanoparticle. I think that's important. In terms of PCC, like pitfalls and things like that, really it comes down to just not knowing what you're measuring. For example, I see a lot of times where people will take the simplest case, DLS versus TEM, right? A lot of people will measure DLS size and then not know which size to use. So DLS, you can get a z-average, there's an intensity weight, there's a volume, there's a number distribution. Which one do you pick? A lot of times people will pick the one that coincides with their TEM measurement, for example, and that's not correct technically. So you should be cognizant of what you're actually measuring using a certain technique. And we published a lot on that. We actually have a paper that's out there, and I could probably forward that to whoever. It's a common pitfalls in nanotechnology. It's actually called that. I think it came out 2 or 3 years ago by our group. And it highlights the more common pitfalls that we've seen in regards to characterization as well as in formulation.
I think our time is up. I would like to thank Jeff once more for a very interesting lecture and for a lot of information to take away. I would like to thank all participants for attending our webinar and for being active participants. And don't hesitate getting in touch with either Jeff or me or any other of the board of the focus group for further questions. And with this, I would like to conclude this webinar and hope to see many of you in July in Valencia at the annual meeting of the CRS. Thank you.
Thank you.